Hey everyone, welcome back to the channel. Today we're breaking down one of the most high-yield MCAT topics, amino acids and protein structure. We'll cover names, abbreviations, and structure of amino acids, the properties of amino acids, their charges at physiological pH, protein structure, how amino acid properties affect protein folding, and why all this matters for enzyme activity. By the end, you'll know exactly what the MCAT expects you to remember about these building blocks of life. Let's dive in. Amino acids are the building blocks of proteins. Every amino acid has a central carbon or alpha carbon atom, an amino group, a carboxyl group, a hydrogen atom, and a unique R group or side chain. That R group is what makes each amino acid different. The MCAT loves to test how side chain properties, polar, nonpolar, acidic, or basic, affect protein behavior. There are 20 naturally occurring amino acids. Let's categorize them in a way that's easy to remember. Number 1. Nonpolar, hydrophobic. There are 9 of them. Glycine, alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, and tryptophan. They are hydrophobic, which means that they do not really like water and hide inside proteins away from water. Mnemonic. Grandma always visits London in May. Pretty phenomenal trip. Number 2. Polar, uncharged. There are 6 of them. Serine, threonine, cysteine, tyrosine, asparagine, glutamine. These can form hydrogen bonds. Mnemonic, Santa's team crafts toys at garage. Number 3, acidic, negatively charged at physiological pH, aspartic acid and glutamic acid. Mnemonic, acidic apple grows. Number 4, basic, positively charged at physiological pH, histidine, lysine, and arginine. Mnemonic, his little army. Besides structure, it may be also important to remember the 3-letter and 1-letter abbreviations of amino acids. Here is a list of abbreviations used for amino acids. MCAT tip. The average molar mass of amino acids is around 110 Daltons. Also, remember, all amino acids are chiral except glycine, and only L-amino acids are used by cells for protein synthesis. At physiological pH around 7.4, amino acids primarily exist as zwitterions, meaning the amino group is protonated and the carboxyl group is deprotonated. Thus, they carry both a positive and negative charge simultaneously. Zwitterion formation is energetically favorable for amino acids in aqueous environments because of these opposite charges. They stabilize the molecule through internal electrostatic balance. They also allow for strong hydrogen bonding with water, making amino acids highly soluble and stable in biological systems. The overall charge of an amino acid depends on the pH of the medium relative to its isoelectric point, or pI, the pH at which the molecule has no net electrical charge. For neutral amino acids, pI approximately equals the sum of the pKa of the amino group and the pKa of the carboxyl group divided by 2. Usually, pI for these amino acids is around 6. It is because the pKa of the carboxyl group is approximately 2 and that of the amino group is approximately 10. For amino acids with acidic or basic side chains, you include the side chain pKa in your calculation. For acidic amino acids, the pI is less than 6, and for basic amino acids, the pI is greater than 6. Why does this matter? Because proteins migrate in gel electrophoresis based on their pI and the pH of the gel. We will describe this in another video on isoelectric point and gel electrophoresis. MCAT tip. Histidine is special because its side chain's pKa is around 6, so it can gain or lose a proton easily. That makes it super important in enzyme active sites. Understanding the chemical properties of amino acid side chains, like histidine's ability to gain or lose a proton near physiological pH, is crucial because these side chains ultimately determine how a protein folds and functions. When amino acids join together, they form long chains that must adopt specific shapes to carry out their roles in the cell. These shapes are organized into 4 levels of protein structure. Primary structure refers to the linear sequence of amino acids in the polypeptide chain held together by peptide bonds. Secondary structure describes local folding patterns stabilized by hydrogen bonding, such as alpha helix and beta sheet. Tertiary structure is the overall three-dimensional shape of a single polypeptide chain formed by interactions between side chains through hydrogen bonds, ionic interactions, hydrophobic packing, and disulfide bridges. Finally, quaternary structure arises when multiple polypeptide subunits assemble into a functional protein complex, stabilized by the same interactions seen in tertiary structure. Together, these levels of organization give proteins their unique shapes and enable them to perform highly specific biological functions. The primary structure of a protein is the exact linear sequence of amino acids connected by peptide bonds. This sequence is determined by the mRNA transcript during translation and is crucial because it dictates all higher levels of protein folding and function. On the MCAT, remember that even a single amino acid change can have major physiological consequences. For example, in sickle cell anemia, a single point mutation replaces a polar glutamate with a nonpolar valine in hemoglobin, causing abnormal aggregation. The chemical properties of the amino acids, polar, hydrophobic, and charged, drive how the chain folds, making primary structure the blueprint for the protein's final 3D shape. Let's dive in a little more. Proteins form when amino acids link together via peptide bonds. This bond is formed through a condensation or dehydration reaction, releasing water. Peptide bonds are rigid, planar, and carries partial positive charge on nitrogen because of resonance. They are always read from the N-terminus to the C-terminus, so peptide bonds are directional. But proteins aren't just chains. They fold into complex shapes. Secondary structure. Protein secondary structure is primarily determined by hydrogen bonding between the backbone atoms of the polypeptide chain. In alpha helix, hydrogen bonds form between the CO group of an amino acid residue and the NH group of another amino acid residue 4 units apart. In beta sheet, hydrogen bonds form between CO and NH groups on adjacent polypeptide strands. Beta sheet could be parallel or antiparallel. Side chains or R groups do not directly form the secondary structure. Hydrogen bonding in alpha helix happens between 2 amino acids that are 4 amino acid units apart. That is, they have 3 amino acids in between. Also remember that certain amino acids cannot form alpha helix. For example, proline, because it introduces a rigid kink in the helix and is often known as helix breaker. Glycine also is not a very good candidate for alpha helix formation because of the presence of tiny hydrogen as its R group. It has too much conformational freedom. Beta sheets are formed when stretches of a polypeptide chain align side by side, stabilized by hydrogen bonds between backbone carbonyl oxygens and amide hydrogens. There are 2 types of beta sheets: In a parallel beta sheet, the adjacent strands run in the same N to C direction, producing slightly angled hydrogen bonds that are less stable. In contrast, an anti-parallel beta sheet has alternating N to C orientations, allowing nearly linear hydrogen bonds that confer greater stability. The connectivity between strands differs in these two arrangements. Anti-parallel sheets are often linked by short beta turns, in which four residues create a tight 180-degree reversal stabilized by a hydrogen bond. Parallel sheets, however, usually require longer crossover connections, sometimes looping nearly 360 degrees to align strands running in the same direction. Proline and glycine frequently appear in beta turns. Proline introduces rigidity, while glycine provides flexibility for sharp bends. Together, these structural features enable beta sheets to fold compactly and form the stable cores of many proteins. Tertiary structure: 3D folding of protein driven by hydrophobic interactions, disulfide bridge from cysteine, hydrogen bonds, hydrophilic interactions, and ionic bonds, or salt bridges. Quaternary structure: multiple polypeptides coming together like in hemoglobin. The polypeptide units are held together by combination of same forces involved in forming tertiary protein structure. For example, hemoglobin with 4 subunits. It is important to remember that not all proteins have quaternary structure. Key MCAT connection: hydrophobic interactions drive folding. Nonpolar residues tuck inside. Polar and charged residues face the watery environment. Now, why does all this matter? The structure of a protein determines its function. If a protein misfolds, it can lose function. Think Alzheimer's or prion diseases. Denaturation by heat, pH changes, or chemicals breaks down these structures, usually destroying protein activity. Enzyme activity depends on the active site, which is shaped by amino acid interactions. The MCAT loves to ask, if you change an amino acid from hydrophobic to charged, what happens to folding or activity? That's where this knowledge comes into play. Let's wrap it up. Amino acids differ by their R groups. Side chain properties: polar, nonpolar, acidic, basic. Control folding and interactions. pKa and pi tell us about amino acid charge at different pH values. Protein structures are built from primary to quaternary, with folding driven by hydrophobic interactions. Enzyme activity relies on these structural principles. If you nail these concepts, you'll be ready for MCAT passages on amino acids, proteins, and enzymes. Thanks for watching. Be sure to like and drop a comment on which MCAT topic you'd like me to cover next.